Well, hello. My name is Francisco. I'm going to be showing you my model of a skin cell to begin with, followed by a couple other ones after that. But let's, I'd say maybe let's go from top to bottom to make it a little bit more easier and for things to make more sense. Up here, we're going to start off with the epidermis, which is more so the dermal papillae, I guess. And then the top part would probably be the hair shaft that pokes out, and that's, external and things that we can see. Um, the actual follicle itself is called the hair follicle, as I just said, and then the bottom would be called hair bulba. Um, this pink stuff that I have around here, I use to illustrate the sebaceous, what's the proper word? Sebaceous gland, which usually get attached to the hair follicles and stuff. And to the right, I have the erector pili. Kind of just memorize stuff here and there. Sebaceous is soft, erector, it is erect to the actual hair follicle. Hair ball, we did that. We also have sweat glands. This would be the eccrine sweat gland. I tried to fit another one, but I couldn't fit. But it's another one called the apocrine sweat gland, and yeah, they're just found all over our body. Some areas have more than others, and some sweat glands produce sweat, obviously, that come with an odor. And all of that would be found in the dermis part of our body. So we did epidermis, we did dermis. I'm trying to see if I forgot something. The eccrine level, and the apocrine, which I don't have. And then going down to the hypodermis, we have what is all these little pink stuff everywhere on the bottom, and that's the adipose, which is basically just fat stored in there, for energy. And yeah. And then we also have the Pacinian. In all of that adipose, we have the Pacinian corpuscle. And one way I try to remember that is by saying like Pacific Ocean, like it's really far away out in the way. So I try to remember that. So Pacinian corpuscle with Pacific Ocean, which to me makes sense. So once again, we have the epidermis, the dermis, the hypodermis, and a couple of things that I could fit in there. All right, my next tissue, it's gonna be the nervous tissue, and obviously for that, I'm gonna pick a neuron, which makes the most sense. I have my notes here just so if I have long pauses because I'm trying to read some of that and remember some of that too. So let's start from the far left, which to me would make the most sense. The whole thing itself is called a soma, which is a cell body. These little points at the end are the dendrites. Hopefully I'm saying that right. The dendrites on the side, which are receiving signals, and ready to go off. But we have those in the middle, the pink that I use, that is the nucleus of the soma itself. Okay, let's keep going. In the middle, between here and here, we have the axon, which is this trunk-like thing that is connected to the myelin. Like I just said, the myelin sheath is all this blue that I have leading up, which is basically like the skin, a nice protective layer that goes over the axon, and the cell itself, the neuron itself, just to keep it safe. And yeah. All right, next we have these orange dots that I have in each of the myelin, and those are the Schwann cell. After that, in between the myelins, we also have, which are nodes. They're just nodes that carry everything, and there's spaces between the myelins. So think of this as basically the protective layer. Between the protective layer, there's little gaps and spaces everywhere that just help fluctuate that transmitter, I guess, that signal that's being received here to come out here. So it's little prints. It just helps it speed up, helps it go nice and smoothly. Once we get to the end and the signal has, been transmitted all throughout, we get to what this is, the whole thing, the axon terminal. Up in the axon terminal, we have between the axon terminal and the next neuron, basically the next dendrites, it would be the synapse, basically. So yeah. All right, my next tissue is going to be the connective tissue. And for the connective tissue, I had a couple options like blood and compact bone and stuff, but I chose the adipose because I feel like we've used that plenty of times in other parts of, tissue. So for this, we're gonna start with the whole thing being wrapped in a cell membrane. The blue outer layer, of this right here, of the adipose, is the cytoplasm, which is just plasma around there. The pink itself is the fat reservoir where the adipose gets its shape. The green over here we see is the mitochondria, and the orange over here is the nucleus of the adipose cell. We see some brown over here. That brown is kind of more spread out, obviously around here, around there in the actual cell, but that's the Golgi apparatus. All right, the adipose is usually found beneath the skin in the hypodermis layer. We can find it around areas like around the organs, in between muscles, and also the breast tissue. It's very helpful in storing energy in the form of fat, obviously, and it makes a good cushion for our internal organs. All right, take two. Let's say epithelium. For my epithelium tissue, I picked the simple columnar only because I thought it would be, like it says in the name, kind of simple. Let's start up here on the top where we can see these hair-like follicles on top. I tried to represent that by just using a pencil and scraping it. That is called the microvilli cilia. They're just hair-like follicles surface that's on top of this, basically. And then each cell has its own cell membrane, which kind of separates them, distinguishes them between each column. The pink stuff itself is called the goblet cell. The goblet cell, and then the green inside the goblet cell is the nucleus. On the bottom, we have the basement membrane. Kind of very easy to remember. We have follicles, hair, like top of your head, so that would be cilia. And then the bottom basement, that's underneath your house, basement membrane. So pretty simple. All right, for muscle tissue, I picked skeletal muscle. Let's start off with over here. I made strands of muscle fibers, with the blue being the nuclei and striations. So I try to make little lines, striations, just to, I guess, imitate the fact that they have little ridges on them. Now, I kind of went a little overboard in my opinion and tried to show you a bigger picture of it, but this is the cell itself. If we were to get a bundle of muscle fibers and wrap it in endomysium, we would get fascicles. If we were to get strands of fascicles and set them up just like this, and then wrap them in Pimicium. I feel like I'm butchering that. Perimycium. We would then get something. We would get the muscle bundle. So it would look kind of like this, where it's just strands of stuff. Just pretend that's covered up. I should have covered that up. The points, the edges are a little bit pinched in, and then the body itself of the muscle. Now, if we were to cut that in half, we would give little examples of this, where this would be all the perimycium, with the fascicles being these circles right here. And then we will also have like arteries and veins and stuff all throughout, but this would be like the bigger picture. So we go from this to fascicles also being bigger to something like this, cut that in half, and then we would get that.